C-peptide is a small protein made of 31 amino acids. Although it was previously thought to be an inert byproduct of insulin, studies focusing on C-peptide have shown that it is an incredibly important biomolecule. C-peptide connects the alpha and beta chains of insulin in the pro-insulin molecule. The C-peptide chain is cleaved from the pro-insulin before the alpha and beta chains of insulin, connected by disulfide bonds, are secreted from beta cells. Typically, C-peptide is secreted in equal molar amounts as insulin in the blood. Type 1 and type 2 diabetes can be distinguished by measuring the C-peptide to creatinine ratio in the urine. A reading below 0.2 nanomole per millimole would suggest that a person has type 1 diabetes, because low C-peptide levels indicate the absence of insulin production. A reading exceeding 0.2 nanomole per millimole would suggest that a person has type 2 diabetes, as elevated C-peptide levels indicate excessive insulin production due to insulin insensitivity. The exact pathway of C-peptide in the body still remains unclear. However, many scientists have proposed potential interaction pathways. One model suggests that the binding of C-peptide to a G-coupled protein receptor starts a cell signaling cascade. The binding causes intracellular calcium levels to rise, and this activates the endothelial nitric oxide synthase. Endothelial nitric oxide synthase produces nitric oxide as a signaling molecule to activate protein kinase C and mitogen activated protein kinase. These kinases in turn activate sodium-potassium ATPase. Sodium-potassium ATPase is a plasma membrane cation pump whose activity is essential for the normal functioning of the body. This enzyme is responsible for regulating cell volume, sending signals and coordinating cell activity, and maintaining concentration gradients between the cell cytoplasm and surrounding environment. C-peptide administration is positively correlated with the sodium-potassium ATPase activity, and therefore complications associated with reduced enzyme activity are a concern for people with type 1 diabetes. Synthetic insulin that is administered in insulin therapy via injections or by an insulin pump contains only the alpha and beta chains of insulin. Therefore, people with type 1 diabetes have no source of C-peptide. It is thought that some of the long-term complications of type 1 diabetes, including decreased blood flow to microvascular tissues, kidney failure, heart disease, dysfunction of retinal cells, and diabetic peripheral neuropathy might not only be caused by extended periods of hyperglycemia, but instead might be due to the lack of C-peptide. Therefore, C-peptide administration and therapies are being extensively studied in order to see if C-peptide could be used as a treatment to reduce long-term complications. One group was interested in how C-peptide influenced sodium-potassium ATPase in kidneys, and found that C-peptide prevented the decrease of sodium-potassium ATPase alpha-1 subunit in rats. By using real-time PCR with a fluorescence thermal cycler, the Nordquist group quantified mRNA expression for the sodium-potassium ATPase alpha-1 subunit. 4 groups of rats, including normal, diabetic untreated, diabetic insulin treated, and diabetic treated with C-peptide, were compared. The data shows that there was an observable decrease in mRNA expression levels for the untreated and insulin-treated rats. However, there was no observable decrease in expression in the rats treated with C-peptide. Studies in humans have shown that C-peptide activates a mitogen-activated protein kinase to increase enzyme expression and transcription. Therefore, it is possible that in this model, the C-peptide also increases the mRNA expression of the catalytic subunit. Additionally, immunohistochemistry was performed to observe the localization of the sodium-potassium ATPase alpha-1 in rat kidneys. There were localization signals observed in both the cortical distal tubules and medullary collecting ducts of the control animals and the diabetic rats treated with C-peptide. Yet, the untreated and insulin-treated diabetic rats showed a decrease in localization in the medulla. Overactivation of protein kinase C leads to internalization of the sodium-potassium ATPase. Since C-peptide can prevent overactivation of the kinase, this may explain why the rats treated with C-peptide did not show decreased localization of the cation pumps. These results led to the conclusion that C-peptide is responsible for preventing decreased expression of the alpha-1 subunit, and that C-peptide is important for the regulation of the enzyme. Although this study demonstrates that C-peptide administration may be able to reduce complications, the rats had diabetes for only a few weeks, and therefore may only have minor complications compared to the complications a person would develop after having diabetes for many years. Therefore, it will be crucial to design studies that compare C-peptide's ability to reduce a wide spectrum of complications. Also, based on the data, it is interesting to consider whether complications could be prevented by C-peptide administration starting at the time of diabetes diagnosis. Furthermore, biochemical pathway details of C-peptide are still being debated. It is thus essential to continue pathway studies in order to understand how and why complications develop and to aid in the design of potential C-peptide therapies.